Start. Okay, welcome. Good morning to everyone. We continue our course on stochasticity. Thank you for those people who still survive. It's the fourth day, so that will be the lecture number 8, and 4 more lectures left. So to remind you, we are continuing to discuss stochasticity, and at this moment we're doing examples. We're trying to learn by examples. Today will be another biological system for which application of stochastic models seems to be successful. Okay, so this is a stochastic mechanism of cell size regulation in bacteria. So you all had a biology in high school, right, or something, right? Did you force you to look at microscope at cells? Did you? Good. So maybe you remember there's a beautiful, they really look beautiful. This is the blood cells look like. Under pretty good microscope, this is the blood cells look like this. This is the human skin cell, look like this. And remember, cells are what we remember. Cell is the main building block of all biological systems. This is onion cells. When you cook onion, if you break the cell, there is a compound which produces and make you cry. This is inside of the cell. This is the plant cells under the microscope. There's also bacteria, beautiful pictures of bacteria. This is E. coli bacteria. This is Bacillus subtilis bacteria. So there is a striking observation. I hope you already noticed what I'm showing you, that while for different organisms and maybe different tissues, cell sites are very different shapes and sizes, for a given organism, given tissue, they're very similar to what we. So you see they're remarkably similar in sizes and shape, right? Let's go here. Remarkably similar. There is some deviation, but they're remarkably similar between each other. Okay, this is so strong that this is still the best test for cancer. So God forbid if somebody asks to do biopsy, that's what they do. They take essentially look at the microscope, that's what they do, and any deviation from cell size uniformity is still one of the best and cheapest markers for cancer, right? There's a lot of development, people trying to do markers, expensive things. This is as cheap as possible. Just look at microscope, count the sizes, see the size distribution. If it's broader than something, it's a sign of disease, cancer. And again, it's another indication of unusual precision in biology, right? Cells are very uniformly distributed in size. Good. Again, this is something that we mentioned. This is a consequence of homeostasis. Homeostasis is this tendency to maintain constant biological properties. We already talked about it several times, right? So again, it is believed that keeping cell size almost constant provides the most efficient functioning for biological systems. But the question comes how biology can do it. What is a possible mechanism to do it? And it remains a source of debate. For people introduced the concept of cell size checkpoint. That was proposed for mammalian cells. And in a simple language, the idea is that there is a devoted biochemical pathways, devoted reaction channels for maintaining the cell size. And some information has been obtained, but it's still limited. At the same time, bacteria, simpler organism. It's not the simplest, it's still complicated. But it's a simple organism where people really studied bacteria quite well, especially E. coli bacteria, they studied that probably beat to them to the death, right? So they checked for these bacteria, and there is no cell size checkpoint. In other words, there are no devoted biochemical pathways which leads to cell size, to very uniform cell sizes. So the question is, what is the mechanism of cell size control in bacteria? And people generally propose two types of ideas how bacteria can achieve the same cell size. One set of ideas called sizer models. sizer idea. And the idea here is very simple one. So this is the number of divisions, this is the time, okay. So they say, if you initially start with a very small cell, the black one, right? Then somehow in mechanism of bacterial, they actually add a lot of size. So that after the next division, your molecule will be increased. And then if your molecule has a long size, you add a little. After several these kind of dying fluctuations, you will reach a stationary state. So you understand, the idea is if originally at the birth cell is too big, then somehow biologically you add a little. But if you originally at the birth you are too small, you add a lot. As a result, after several divisions, you will get almost constant size. So this is called sizer model. Somehow bacteria can size over new size model. There is a second type of ideas, and these are called adder model. And here the idea is different. Here the idea is that on average, between every division, cell adds approximately the same volume. And if you continue the exercise, you will actually see that you reach some stationary size eventually in the so-called adder model. Okay.
Professor, when you said this added size, is this sometime after division?
Right, okay, so let's repeat. So what's going on? Time zero, this is your original bacterial cell, right? Then after the next division happens, you add this amount, cell divide, right? And this will be a newborn cell after division of this in half. Black is a newborn. The red is added between divisions. Newborn added. Okay, maybe my drawing was not successful. So you start, you grow, then you divide again. You start, you grow, you divide. Then eventually you will reach a stationary state. And there are two different limiting cases. In first cases, you add a lot if you're small, you add a little if you're large. And then after again, several of those things will lead to stationary size.
But it's add and then divide in half.
Yes. So you increase, you divide, and the new cell is about half of what is the maximum range. Okay, there is so as you probably know, significant advancement in this field came out with experimental development.
How this addition value or divider value decided?
Good question. That's what they think, but they don't explain why. I will explain you how something comes out from a more microscopic picture. They do it phenomenologically, okay? They do it phenomenologically without explanation why. Good. So there is a significant development came with this experimental advances, so-called single-cell microfluidic device. It's in the language, in the slang, they call it mother machine. So so the idea is very simple. They had a device which has many channels, and then they added something to bacteria, so bacteria become fluorescent. So then you can see the bacteria. The video might work here, but it will see. It might work. But this was a very convenient setup because you were able to quantitatively measure things. You could measure quantitatively things, and you can see this bacteria. I hope it will work here. Good. Yeah, this is a picture. Do you understand? So imagine there are many channels, and the bacteria start to grow, and then there's a flow over there. So you simultaneously look at like 96 these channels, all start at the same time. You simultaneously can measure sizes. I hope you can see there is a bacteria sizes, and you can also measure dynamics once the bacteria is divided and so on. Can you see it well? Yeah. In theoretical studies, huge progress in experiments drive significant achievement, significant progress in theory too. And that's what the case. That's because numbers here. There's a quantitative measurement. You can test your theory. Okay. Good. So when they did this measurement, they studied this bacteria. They studied this E. coli and B. subtilis bacteria, and what they looked over there, it's a change of size for different conditions, in glycerol. Okay, so this is simply saying different food conditions, they check the average size of added things. Okay, so they measured here how much is added between divisions at different food conditions, and it's the same. Well, for different food conditions, not the same, but for given food condition, it's the same, so that you can put everything in one curve. Okay, so let me give you maybe a little bit simpler explanation what they did, right? If you always eat this Big Mac, many people will be like this. Okay, but they will be all of them will be like this. Okay, so if you really know what to do, so this is Indian food, and that's what will be like. Wait. When I did it, after I edited, I actually turned out to do this election photo. So people in India lining for elections. I'm sorry, but I didn't know it. But this is the same point here. You understand, right? There's nothing wrong here. Bacteria, depending on the food, adds different amount, but it adds the same amount for given food. Okay, which means this is adder principle. Remember what I told you a couple times ago? This is adder. This is the idea that between divisions you add approximately the same amount. Okay, this picture says that between divisions you added approximately the same amount. So, which was very nice story. Huh? Yeah. Okay. Good. This is made in America. Perfectly understand it's America, right? Good. So, what is the current view? How people think about this? People think about this, and the way to explain it currently, before doing stochastic modeling, they use the idea is that there are two conditions must be satisfied for this picture to occur. One condition is called balanced biosynthesis, but this is biologist language. Let me try to use it in a simpler language. Again, it's not me. Okay, so balanced biosynthesis means that proteins, those important proteins that are responsible for growth and division, are created on time scales much faster than the time scale of growth and division. For in the language of physicists, this is called adiabatic approximation. You had physics at least, most of you, right? I know you hate chemistry, but in physics you get it, right? There's very common adiabatic approximation. For example, nuclei and electrons, nuclei moving very slowly, electron moves much faster. So the very successful quantum mechanical description, which is so-called adiabatic one, which says electrons much faster. The same thing story here. The proteins responsible for growth and division created much faster than time scales for growth and division. This is called balanced biosynthesis. This is very reasonable. This is not very reasonable, the assumption of threshold. It's not that it's not observed already. I showed you many times we have in biology thresholds, right? But the saying threshold is it's not like to solve the problem. Okay? It doesn't explain where a threshold comes from. So you need to have a more microscopic explanation where a threshold comes from. Okay, good. This comes to stochastic hypothesis. So again, so remember, this is an interesting story. You have effectively very deterministic outcome, right? And so my idea is the idea that I'm suggesting that stochastic picture can explain this. This is our hypothesis that cell-size regulation it may be due to the coupling of several stochastic processes. Okay, and as soon as you assume stochasticity, there is no need to think about threshold. Threshold behavior should like naturally effectively. This is the main idea, and let's do the model. And we do again essentially one-dimensional model. Why we do one-dimensional model? That's because the bacteria effectively grow mostly along one dimension. Okay, and it's a simple model you can do. So the model is very simple. You associate the size of bacteria with some discrete variable, which might correspond to the, for example, number of proteins responsible for growth. In E. coli, so protein called SDSZ is known to be responsible for growth. Okay, so it's a discrete variable, but it might correspond to number of proteins, and simultaneously this number of these proteins correlates with the size. So this is discrete, again, discrete state stochastic model. We can assume that again the cell proteins responsible for growth division is growth and division perform much faster. And this is very reasonable for typical conditions when nutrients are sufficiently available. And we can say that this protein is the most important protein that proportional to this number and this discrete variable. This is consistent with what I told you about this balance biosynthesis. Yeah, this is extremely simple, naive stochastic model. Two processes coupled, but coupled, properly coupled. Okay, they're in some sense orthogonal to each other. And you can say that the growth happens with rate lambda, and lambda is rate constant. And this division rate happens with rate k, and k is division rate constant. The important thing is the division in principle can take place for any size, but as I will show you, there is only preferred sizes where it's happening. But in principle, it can happen anytime. There is no need for threshold; it will come naturally. Okay.
Professor, is n the number of molecules of those particular proteins?
Yes, but simultaneously you can think about n as size. It's correlates with size, length, for example.
And those intensities are increasing because the number of molecules of protein are increasing?
Right. Oh, it's okay. That's a good question, which I didn't explain. So imagine that I have n proteins responsible for growth. They're responsible for growth. It means that they're adding something to cell wall. If there are more of them, they can add more. So the growth should be proportionally to the number of these proteins, right? The same for the death proteins. The more of this death protein, the higher probability of death. Division, pardon, death, division. Okay, so that's the idea. It is quasi-chemical picture. Good. So we can look at this model differently. There are two ways we can do it. Let's try to do the simplest mean field approach. Okay. So again, this means average size, bracket average size. So I can write equation for evolution of this temporal evolution of this average length, and it has these two terms. Now let's try to discuss these two terms. The first term is easy. This is just a growth, right? So if I wouldn't have division, cells would grow exponentially. But there is a division, and the division has a complex relation. Let's try to explain. The first, K, this is the rate of division, Kn, okay? Why is that coefficient 2 comes? That's because you are, after division, you're creating two new cells. Remember, one mother creates two daughter cells, so that's why you do 2. And then you're also eliminating lengths n over 2. You're dividing, for the assumption here implicit that you're dividing equally in the middle. There are organisms like yeast where it's not symmetric, but for simplicity, let's forget about it. So this is the equation. This is the rate of division coefficient that comes from creating two cells, and this is the length that you are eliminating. You can solve this equation. Very simple. It's essentially a combination of exponents, and you can even present it in this dimensionless language, as people in mathematics like to do, right? So there is no number here. And here is experimental data. So the point of experimental data, the light is the fitting of this theory, extremely simple theory. And it works. And please note that sometimes size of newborn cell could be larger, sometimes could be smaller, but eventually after several divisions, you are approaching a stationary state. And this should show you how this was done. See, this is from the experimental paper, how these measurements were done. I hope you can see this is much better than what before. You can see cell divides, right? Good. So first of all, excellent agreement with the data, essentially without fitting parameters, because the only parameter which was in the theory was this growth rate, which you can get independently. You can get independently. So, and then this also shows that bacteria can reach this homeostasis conditions very quickly, even if it's originally deviate quite strongly. So this shows you why the system very quickly goes to very narrow distribution in this picture. Okay, good. So that was the mean field all time behavior. Yes.
Sorry.
Mm-hmm.
When that event happens with the green arrow with the K or 2K, what is going to the state? I mean, I understand you go from 1 to 2.
Right, right. Okay, so this is this is your original cell. It grows 1, 2, 3. But then it's division. That's it. Disappears from the system.
Oh.
Okay. So I can look at a little bit better analysis, but we have to go for stationary limit. And the stationary limit, I can do something. At every state of the system, this is state of the system, I can define probability to divide. It's very simple. It's a rate of division of the total rates of the system, and it's given by this expression. And important that the stochastic picture, this probability is always constant and independent of cell. You can calculate on this quantity. For example, this is a probability to divide exactly after adding exactly n units. Okay, if you start originally from the length and zero, and that's two possible graph of the same function, which is here the growth is faster, here the division is faster. Again, this expression is physically obvious, right? So there was no division on n events. On events n plus 1, there is a division. Good. But you can use this quantity also to calculate average added length. You can calculate what is the average added length between divisions, right? Q of n is the probability of division exactly at n, right? So if you do this average, and you're getting amazing result. It doesn't matter what you do, how or you start, you always add the same amount of length in this stochastic picture. So you see stochastic picture consistent with experiment. You always add the same length. And this length is essentially balanced between growth rate constant and division rate constant. Okay, so you're getting immediately Adler principle observation, and this average added length is equal to the average cell length. Again, stochastic model gives you very deterministic picture. I hope you can see. Okay, and despite the fact that division can happen anytime, the most preferred division happens at when you add this length lambda over k. Yeah, so you can calculate everything because the model is relatively simple. You can analytically calculate any possible moments, and what I showed here. So essentially, you will have a distribution of sizes, right? Of added sizes between divisions. This is average one is usually peak, and you can see that this is the larger the growth rate, the more narrow the distribution. Okay, so that was a spatial picture. The advantage of stochastic model not only that you can explain the spatial very narrow distribution, the stochastic model can explain dynamics. Remember, it was the spatial narrow distribution as well as the dynamics of division. So you can explain division dynamics. And the way to do it, first passage. That's why we talked about first passage. It's not the only way to do it, but first passage is very convenient picture. So let me repeat again. So I'd like to know the time. Suppose I start in state 3. And suppose it's my time 0. That was my original size. And I would like to know what is the dynamics to divide exactly at n. Okay. And clearly, this is the first passage problem. This is when for the first time your system disappears from this thing. Yeah, so you can solve the things here, first passage again. I don't need to show the equations here. And you can calculate this probability of adding exactly length L. You can also calculate this mean interdivision times, which is exactly mean first passage times. Okay, you can calculate them analytically. And you can see the experimental data. Again, this is the model, and this is the data, the same data that I showed you. See, that works really, really well. But not only it works well, so it's not so important. It's good if your theory can explain experimental data, but more important that it can provide microscopic explanation. And this picture can provide more microscopic picture. So what we see here, so this is a rescale generation time between divisions, but rescale in dimensionless units, time between divisions. And this is rescale new Bohr length. So it's a length of cell size, bacteria when it's after it was born. And there is decrease. The question is why there is decrease? Why there is decrease? And the reason is actually very simple. Let me go back to the model and you'll understand. So the point here is, if I start with this size and I divide it over here, the dynamics at the beginning is relatively slow overall dynamics. So it will take me longer to add exactly the same length. But if I start at larger lengths already, and I still need to add the same length, it will be faster dynamics. So the model explains why this observation, why it's going down. That's because if your newborn length is already larger, the overall dynamic in the system growth and division is faster, and that you can add exactly the same length sooner. Hope it's clear. Okay, so this agrees with experimental divisions. You can calculate also all possible moments. You have distribution of these types of divisions, and it works reasonably well in comparison with experiments. So this is advantages of stochastic theoretical approach. I told you, I'm selling you this. Now it's a card. This is a really convenient method to explain things here. It's a physically very simple picture. Okay, simple model. You can solve this analytically, and you can solve it analytically for both cell size distribution as well as a temporal distributions. So essentially, this reliance on stochastic mechanism eliminates the need to use concept of threshold. Threshold comes naturally. The models are coupled in the following way that the most probable division happens when lambda over k is added. When you add the same lengths, that's the most probable division. It stochastically will fluctuate, but the most probable will be a specific length. So it's a threshold behavioral, deterministic one, but obtained by stochastic means. So excellent agreement with experimental data and many other things. Okay. So again, as I tried to mention here, this almost deterministic cell-size regulation can be achieved by proper coupling. I'm trying to do pictures like this to show that these stochastic models in some sense orthogonal to each other. They're orthogonal to each other, and although each of them will, as we talked in our first lecture, will be a noise, right? It's like a chemical reaction. But because of this proper coupling, this noise kind of cancel each other. That's the story. So stochastic processes, despite being random, if you couple them properly, they can cancel, effectively cancel the noise. Yes, and the average length is determined by the balance of growth and division. This is essentially the better illustration of what I told you. Roads in India, right? Intersection. Despite everything looks chaotic, traffic flows, right? Eventually, traffic flows and so on. This is a similar story, despite the fact that each of these processes along the growth as well as over there is random, stochastic. The overall outcome is almost deterministic, right? You will essentially drive with a more or less constant speed. Yeah. So this is the story. Again, I'm trying not to give very complicated stories, so it's a relatively simple story. But I thought you would really appreciate it. In my opinion, it's one of the best examples that stochastic picture works quite well. Right? So it's based on hypothesis that coupling of several stochastic processes, proper coupling, okay, that's what I'm trying to argue. Proper coupling can lead to effectively deterministic outcome and can explain these things here. So we have a very simple theory. We don't need thresholds. Threshold comes naturally. This we can have a good description of almost everything, and importantly, we can even explain these trends, experimental trends, quite well in this picture. And again, this is the most important things, which should be true in principle, not only for this problem. The idea that I'm trying to promote biological processes, as we mentioned, that in most cases precise, almost deterministic, despite the fact that they're based on stochastic processes. This example shows you can couple stochastic processes, which are inherently random, and still produce almost deterministic outcome. It remains checked, is it true or not? You know, biology is much more complicated than we know, but this is the story. Okay, so any questions? Mm-hmm. Yes, please.
Yes, so you said that the idea was that stochastic processes properly coupled can lead to a more or less deterministic outcome.
Yes.
Is the more or less deterministic outcome captured by the spread of values being small?
Yeah. So it's essentially, let's come back here. Okay, so here, so this is a system, right? You can think about it as a size, right? And it's randomly changing. It's increasing, and it's randomly dividing. But you see, if your cells are too small, the rate of division will be too small, unlikely to see. If your cells are too long, the rate of division will be very long, so you don't see the cells anymore, which this argument shows that there is some narrow range of sizes at which growth will be bounded by division, and that's what you see, right? Despite the fact that growth is stochastic, division is stochastic.
Is this the sigma you had a few slides back?
Six sigma is one of the things. Yeah, it's essentially, yeah. So we have some. Yeah, we can calculate everything, but the point here is this picture that there is an average added size is always the same independent of where you start. That's because you are too short, will not divide, too long, do not exist. There is some optimal length which corresponds to balance between growth and division. Any questions?
So the idea is like you have bacteria and then food, and then they are actually developing. What will happen if they will compete? Let's say maybe multiple bacteria will compete for
That's a
another thing.
much more complicated thing, but in principle, again, stochasticity can capture it.
Is it like let's say assume two kinds of bacteria? Are they going to always have the same steady state level?
It will depend on who is eating food faster or something like this.
Okay, but there may not be definitely the length size of almost all.
It might not only complexity, also even let's say with the case of one bacteria, right? So this is by the way, this is conditions when you have a lot of food. Always have McDonald's with a hamburger, right?
Yeah.
But you can understand that somewhere might not be enough food. If not enough food, the bacteria can really change some behaviors. And this kind of single model will not work. You will need to have several possible states, and that creates a lot of things. Because when people believe that cancer cells, for example, do not follow this adder principle. For example, there is a different thing. But cancer cells are too heterogeneous. There are too many different types, but there are observations that they do not follow the simple picture. Mm-hmm. Any other questions?
No, let's thank the speaker.
Right,